We're going to continue with Ray Price. He is, again, the Senior Director of Business Development at DiscoverX. Title of his presentation, Advances in Research Tools to Accelerate Drug Development. Ray.
Thanks so much for coming and for the BioProcess International for inviting me and for the opportunity to speak to you today. My name again is Ray Price, and the agenda is to briefly introduce the platform technology, the BioSeek platform of primary human cells, and then give three very brief examples about how we've worked with biologics, biosimilars, biobetters, applications of the technology there, immuno-oncology models, and then combination studies. So our technology platform is really built on three separate pillars. Again, we work almost exclusively with primary human cells. There's some exceptions in oncology where we may work with cell lines, but we built more than 50 different primary human cell systems. We build these into disease models using growth factors or cytokines to model a disease environment, and then look at how drugs change biomarker responses in these systems. And what makes it particularly compelling is the ability to look at and compare profiles that we generate with a reference profile database of more than 4,000 compounds that are approved, in development, drugs that have failed in the clinic, biologics, tool compounds, etc. And we can use this database and information to build informatics tools to be able to predict from the signatures that we generate in our profiles clinical outcomes. So, for example, P38 MAP kinase inhibitors have a particular signature that correlates with the skin rash that they produce. Our systems are built, again, to be as physiologically relevant as possible. Often this means generating co-cultures of cells, endothelial cells and PBMCs, cells that produce factors and cells that respond to them, allowing us to recapitulate what goes on in vivo or in the clinic. And these are disease-relevant assays as well. These are human primary cells. Often this is the first time that a client's molecule has been tested in a primary cell model. Our systems are built for drug discovery, so they're quantitative, robust, and reproducible. We work almost exclusively with protein-based biomarker readouts, at least in our standard systems. We can do, though, microarray profiling, phosphoprotein, flow cytometry, etc., and build out custom readouts depending on the needs of the client. The systems are also built and validated with benchmark drugs and compounds. So as we build a system, we look at standards of care or drugs that are physiologically relevant for that particular application and make sure that the systems respond in the same way to what's known about how they respond in the clinic. Right? And again, we build on this based on the reference drug database, the ability to mine that data for using informatics tools, and the comparative analysis to this large library of test compounds. So, same thing, different way. Again, we build systems with primary human cells, a range of different cell types, endothelial cells, immune cells, macrophages, fibroblasts, epithelial cells. Many of these we put together in co-cultures, so endothelial cells and PBMCs, and then stimulate them to model a disease environment, right? Th1 cytokines to model an inflammatory environment, for example. Typically, we put these together in a broad set of systems or a panel, and this is what we call a BioMAP profile. That's what's shown here. This is an example with Prednisolone, glucocorticoid receptor ligand, or a steroid. Across the top are 5 of our BioMAP systems. From left to right, we have coronary artery smooth muscle cells, 2 dermal fibroblast models, either resting or stimulated with cytokines and growth factors to model a wound healing or psoriasis environment. And then at the far right, 2 co-cultures of endothelial cells and PBMCs, stimulated either with LPS to induce a monocyte response or superantigen to induce a T-cell response. Across the bottom, probably not legible on the screen here, but these are, again, mostly protein-based biomarkers. These are selected based on their physiological and translatable activities, both in clinic and for translational researchers. We also do measure cytotoxicity and proliferation in these systems, so if your molecule is overtly cytotoxic, we can detect that in our assays. The y-axis here is the log expression ratio of drug-treated cells to DMSO control. So we normalize essentially our systems to a DMSO-treated set of cells, same cells and stimuli. Therefore, any increase or decrease above or below this line, this horizontal dashed line, are changes in biomarker responses specifically induced by the drug. This is an example again for Prednisolone. So we look also for activities that fall outside of that gray shaded area. Hopefully you can see that, this 99% significance envelope. essentially this is a measure of assay variability. And so we look for changes in biomarker expression outside of that as a measure of statistical significance. And this is quantitative, so here concentrations of prednisolone from 3 micromolar down to 4 nanomolar in green. One thing you can appreciate about the profile is that certainly the magnitude of these changes increases with increasing dose, but the overall shape of the profile is actually quite consistent across this range of doses. And this is what we call then a signature profile for prednisolone. Other glucocorticoid ligands might share some aspects of this, but then when we profile drugs for clients, we're able to match their profiles to this 3,000 compound database to be able to understand and deconvolute mechanism of action, right? Address on-target versus off-target activities, etc. And this can be done, more broadly. Here, what we're showing, or what we've done, is taken one of the profiles that I showed you for prednisolone, and we're representing that by a single point here. So here, for example, in the bottom left are 2 different p38 MAP kinase inhibitors. Either the BIRB796, shading here indicates 2 different concentrations of that drug, or the VX745. We then do a Pearson correlation and look at how similar these profiles are to each other, connect them with a line if they share a Pearson of above 0.7, and the length of the line indicates the overall similarity of those 2 profiles. What this means is that we can identify by their phenotypic profiles that they produce common mechanisms of action of these drugs. p38 MAP kinase inhibitors cluster together, calcineurin inhibitors cluster together, here cyclosporin A, FK506, different mechanisms of inhibiting that, but produce similar phenotypic profiles in our systems, PI3 kinase mTOR inhibitors, and even very broad cell-based activities like mitochondrial electron transport chain modulators. Right? So the ability to distinguish compounds, both single-target selective molecules, pathway modulators, and even very broad cell-based activities. So that's, again, the BioSeek platform. I'm going to go through then 3 examples of different applications of the technology, the first in biologics. And this is a comparison of different anti-TNF alpha antibodies in our systems. So again, the systems are listed across the top here, various primary cells, stimuli present in them. And here we're looking, and again, any increase or decrease above or below this horizontal line, especially outside of this gray shaded area, are changes in biomarkers specifically induced by the drug, either increase or decrease. This is a profile of 3 different anti-TNF antibodies. This is a period here, so these are 1 microgram per mil, selected for the same concentration of Enbrel, Humira, and Remicade. Right? And certainly these molecules differ in some minor aspects in where these arrows are in terms of the potency on select activities. But in terms of positioning a biosimilar or bio-better, right, the ability to very broadly profile your molecule across a broad set of biology, say pharmacologically, maybe the modifications that we made change the PK of this molecule, but still preserves the pharmacology or its intended efficacy. And we can do that in an even more dramatic way here. Here we've taken, in the red profiles, these are monovalent Fab fragments of Remicade, and then the full-length bivalent fragments in blue, and different concentrations of those under those colors. And what you can appreciate is that the profiles differ in terms of their, certainly they're equivalent in terms of their ability to inhibit TNF, these large peaks here and here, and macrophage activation, right, almost overlapping, but different in their ability to affect T-cell function, right, things like CD69, IL-10, or IL-17 production, right? So one conclusion from this is that these monovalent TNF inhibitors may be less immunosuppressive than bivalent agents, and that could be an important aspect of your clinical development plan, right? Improving on the selectivity or the pharmacology of an existing molecule. Let's switch gears here and talk about our oncology systems, again, very briefly. Here it's the same concept of taking primary human cells, right? So either a fibroblast or endothelial cell as the background sort of stromal or vascular environment, an immune cell component to model that, and then in this case we're talking about a HT29 tumor cell line. So really a fairly robust and complicated model of the tumor microenvironment. We co-culture all 3 of those cell types together, add a drug, biologic or a small molecule, and then look at and stimulate these systems to create a modest inflammatory environment. So T cells are primed but not cycling. and then look at both with flow cytometry to look at immune checkpoint expression in this case, as well as a set of biomarkers that reflect changes in the stromal, vascular, immune, or oncology-based readouts. Right? So the same concept we've been talking about here, we've just introduced a model of the tumor itself. And with the announcements at ASCO in the last couple of weeks, right, interest in this molecule, Pembrolizumab. This is from a clinical pharmacy that we've sourced, anti-PD-1 antibody. And what we can see here, again, across the top are 4 of their BioMAP systems, either the HT29 containing systems on the left and in the middle left here, or on the right side are the non-small cell lung cancer cell line, H1299s, either a fibroblast or endothelial cell containing system. And these are concentrations of Pembrolizumab from 50 micrograms per mil down to 1.8 micrograms per mil. And what we can see, consistent with what's seen clinically, is the increases in biomarkers that reflect inflammatory or immune responses, right? Increases in granzyme B, interferon gamma, IL-10, IL-17, etc. Interestingly, in the fibroblast-containing systems, inhibition of VEGF, we are developing this actually as seems to be a specific biomarker for tumor cells. But the same thing overall is seen in the endothelial cell containing system, increases or potentiation in inflammatory or immune responses. Right? And certainly these systems are amenable to combination studies, right? So combinations of checkpoint antibodies, companies with small molecules developing those, looking to see their suitability in combination with these checkpoint antibodies as well. And that's the example, my third example here. This is a different biology, so now looking at fibrosis, and looking at the 2 compounds we'll talk about, Pirfenidone and Nintedanib, both recently approved in idiopathic pulmonary fibrosis. Here we take either renal epithelial cells or small airway epithelial cells, co-culture those with airway fibroblasts, stimulate them in a way that causes differentiation into myofibroblasts, and then look again at biomarkers that reflect that transformation, things like alpha-smooth muscle actin, N-cadherin, E-cadherins, but also the matrix remodeling that's occurring, things like collagens, MMP9s, and inflammatory or immune responses, cytokine production, and growth factor receptor production. The data is shown here. What we're looking at is Nintedanib at 3 micromolar, this profile in red, Pirfenidone at 5 millimolar in blue. And again, the y-axis is the log ratio of drug to DMSO control. So any increase or decrease that fall outside of this gray shaded area are biomarker changes that reflect drug-induced and specific drug-induced biomarker changes. What we're showing here is that activities seen when we, in both cases, right, reveal an anti-fibrotic signature. So these molecules impact matrix remodeling. They impact biomarkers like alpha-smooth muscle actin, the N-cadherins, and PI1 across multiple systems, right? Many of those are annotated here, the collagens, right? These are components of the mature matrix that the cells lay down. Inhibiting that is consistent with anti-fibrotic activity. We also see differences in terms of the systems that they're active in, but there's still a common impact of biomarkers for inflammation, right? And our hypothesis now is that these molecules, or molecules more likely to be successful if you impact both anti-fibrotic activities and anti-inflammatory activities, right? And molecules that we profiled, both that were effective or clinically successful like these 2, as well as ones that don't, tend to support that idea of being able to impact both inflammation and matrix remodeling activities. The strategy and the design that we've used to be able to do that is shown in this slide. This is a combo elect profiling, so we would profile both agents by themselves. In this case, for example, Pirfenidone and Nintedanib would be profiled by themselves, and then generate a 4x4 matrix of how those 2 drugs interact across a set of 16 different parameters or conditions, right, that are listed here. And then a robust statistical background to look at how the combinations change compared to either drug alone. Are they synergistic, potentiated, negatively interacting, etc.? And these can be anything, a small molecule and a biologic, a stimuli and a test agent, etc. And that data is shown here for the combination of Nintedanib and Pirfenidone. So again, the BioMAP systems across the top here, this is the small airway epithelial cells co-cultured with myofibroblasts. The profile from Nintedanib, this is a lower concentration that I showed previously, is shown in blue, and Pirfenidone at the 5 millimolar concentration in red. And these, what we've annotated here, are activities that are significantly different in the combinations versus either compound alone. And again, looking at biomarkers that reflect both antifibrotic activity, so inhibition of collagen 1, alpha smooth muscle actin, right? Collagen 1 and 3 alpha smooth muscle actin here. So both antifibrotic activities as well as anti-inflammatory markers, things like ITAC, IL-8, right, MCSF, etc. So the ability to model both accurately the biology in low-passage primary human cell assays, but design experiments in a way that makes sense with clinical development and strategies. And so we can address this, from anywhere really in the preclinical or clinical spectrum. So from broad panels that are designed to sort of ask the question, what's happening in the rest of the patient? What activities are covered by a particular drug or inhibited, indication selection, right? Both Diversity Plus or a panel that we've developed out of our EPA ToxCast relationship of predictive tox, so the ability to look at signatures and how they correlate or not with side effects or toxicity. Again, we have a set of panels and systems for focused biology. We put these four systems that reflect T-cell biology, oncology systems that I mentioned, and then fibrosis-related activities. And one can access individual system or even individual readouts, certainly going early in the SAR, lead identification, or hit ID, but also even through phase 4. We've worked with Roche and presented at ACR last year to look at the combination of Actemra with methotrexate. How did those two interact? Is there overlapping or complementary biology that those impact? And that's my last slide. I'm happy to take any questions or comments at this time. Thanks for your attention. I think there's a microphone if you want to use it. All right. I'm sorry? Yeah. That's great. All right. Thank you very much. All right. Go ahead.
So in your investigations, it seems like most of the comparisons that you made, the drugs that acted similarly gave similar profiles. Were you ever surprised if they were structurally divergent, that they didn't invoke other systems like, say, metabolic systems or something like that differentially? Did that ever surprise you?
So it's very common so the question is basically around structural similarities, and right, if you had two compounds that impact the same single target perhaps, but do they have off-target activities or different because of their structure? Yeah. So we certainly have seen that. I'll say it's very common, especially for these broad panels, for us to find activities that are a surprise or that were unsuspected to the clients. So for example, the P38 MAP kinase inhibitor I mentioned, we do see convergent biomarker changes in the models that we have for the skin rash toxicity, but they may have other activities in our immune-based systems as well, or may have differential, and that's a very structurally diverse class and selectivity. Right, yeah. Yeah, so that's right. So, we do, this was selected a little bit based on, right, for simplicity, this is only about 45 or 50 of the molecules that we profiled, but you certainly can do that as well for, looking at structurally diverse molecules, looking at target selectivity or on-target, off-target activities. So, thanks for your question. All right. Thank you.